All right, let's pull back the curtain on the microscopic world that literally builds the food we eat. Today, we're talking about the master builders, the incredible architects behind every single bite: carbohydrates, proteins, and fats.
I mean, have you ever really stopped to think about that? What is it that makes bread so chewy or gives jam that perfect wobbly, gel-like texture? Or what makes butter so incredibly smooth?
Well, it's not magic, even though it kind of feels like it sometimes. The answer is pure science. It all comes down to three super powerful, yet totally invisible, building blocks called biomacromolecules. And here they are, our three food architects: polysaccharides, which you know as carbs, proteins, and lipids, which we call fats. These guys are responsible for the structure, the texture, and even the nutritional quality of pretty much everything on your plate. So let's break them down one by one. Okay, first up, we have the most abundant of the bunch: polysaccharides. Now, you probably know them better as complex carbohydrates. The easiest way to think of them? Just imagine long, elegant chains made of sugar. So the big idea here is that they're polymers. Think about a pearl necklace, right? It's just one bead after another linked together. A polysaccharide is exactly like that, but its beads are simple sugar units, like glucose, all strung together in a really long chain. And trust me, these things are everywhere in our food. Starch is what makes grains like rice and wheat so packed with energy. Cellulose gives plants their rigid structure. Pectin, that's the secret weapon that makes your jam set just right. And gums, they're used to thicken all sorts of things, from salad dressing to ice cream. Now, here's what's so cool about them. Polysaccharides have two totally different but equally critical jobs. On one side, you've got starch, which is all about storing energy for the plant to use later. And on the other side, you've got cellulose, which is like the plant's skeleton, giving it all that strength and structural support. The real secret to how starch works is all in its shape. See, it's made of two parts: amylose, which forms these neat, straight chains, and amylopectin, which is all branched out and kind of messy. And it's the ratio of these two that gives different starches, like, say, cornstarch versus potato starch, their totally unique cooking properties. And here you can really see the difference. Unlike starch, the chains in cellulose are perfectly straight and unbranched. They line up side by side and lock together, forming these incredibly strong fibers. In fact, they're so strong that our bodies don't have the right tools, the right enzymes, to break them apart. And that's why cellulose just passes through us as dietary fiber. All right, let's move on to our second architect: proteins. Now, if carbs are these simple chains, proteins are the complex, three-dimensional shapeshifters of the food world. So, you know how polysaccharides are chains of sugars? Well, proteins are chains of things called amino acids. But the real magic of a protein isn't just in the chain, it's in how that chain twists and folds and bends itself into a very specific, very functional 3D shape. This right here is the absolute key to understanding proteins. Their job, their function, depends entirely on their final 3D shape, their native conformation. If you mess up the shape, you ruin the function. Think about cooking an egg. The heat changes the protein shape permanently. That's why you can't uncook it. And getting to that final shape happens in 4 stages. First, you have the primary structure, which is just the simple, straight line of amino acids. Then, at the secondary level, that chain starts to coil up into basic shapes, like helices. After that, those shapes all fold together into a full 3D working unit. That's the tertiary structure. And finally, in some really big proteins, you get the quaternary structure, where multiple folded chains come together to form one giant complex. It's just an amazing buildup of complexity. And that brings us to our third and final architect: lipids. You know them as fats and oils. These are these serious energy powerhouses and the flavor carriers of the food world. Now, what's really fascinating about lipids is that they aren't defined by a repeating structure like carbs and proteins are. Nope, they're defined by how they behave. And their main behavior? They don't mix with water. That's it. Think about oil and vinegar in salad dressing. That simple property is what makes something a lipid. And their jobs are absolutely essential. I mean, they're our most concentrated form of energy storage. They make up the membranes of every single cell in our bodies. And when it comes to food, they're responsible for carrying all those delicious flavor compounds and for helping us absorb crucial vitamins like A, D, E, and K. So you always hear about saturated versus unsaturated fats, right? Well, the difference is actually pretty simple, and it's all about chemistry.
Saturated fat chains are straight, they don't have any double bonds. But unsaturated fat chains have at least one double bond, which creates a kink or a bend in the chain.
And that tiny little kink has a huge, visible consequence. The straight chains in saturated fats, like the ones in butter, can pack together really tightly, which is why they're solid. But those kinky bent chains in unsaturated fats, like in olive oil, just can't pack together neatly, so they stay liquid. It's molecular geometry in action, and it's why our some membranes stay nice and fluid too.
So, we've met our architects. We know who they are. But the next question is, how do scientists actually study these molecules to, create the future of food? Right? It's one thing to know these biomacromolecules exist, but how in the world do food scientists actually look at them and analyze them when they're way too small to see with a regular microscope? Well, they have this amazing toolbox of really advanced techniques. They use things like chromatography, which is basically a molecular racetrack that sorts molecules by size. They use spectroscopy, which shoots light at molecules to read their unique fingerprint and identify them. And with super powerful microscopy, they can actually take high-resolution pictures of these tiny structures we've been talking about. And this isn't just for fun. This is serious business. By understanding these molecules on such a deep level, scientists can design foods with better textures, make products that last longer on the shelf, and even develop healthier ingredients. This is the science that's driving all the innovation in the food industry. So I'll leave you with this final thought. The next time you sit down for a meal, maybe you're spreading butter on toast, take a second. Think about the lipids making that butter so smooth, the starch in the bread giving you energy, and the cellulose giving a piece of fruit that satisfying crunch. Which of these tiny architects will you notice at work?